Okay, in this session I'm going to go through some of the main methods that are used in cell biology studies for the detection of apoptosis and to differentiate apoptosis from necrosis. So after this session you should be able to propose methods that will distinguish between apoptosis and necrosis in several different situations, both live cell culture experiments, situations where the cells are being fixed, so the cells are dead, so typically we'll use cytospins. We'll look at tissue sections and we'll look at biochemical assays in cell lysates. So just as a reminder, what you've already seen, you've seen that apoptotic cells have condensed nuclei, and we can visualize that with a stain called DAPI. And there are various variations on this, you'll see this later in this session, but the nuclei differ in their shape. And the reason the nuclei differ in their shape is because during apoptosis we've got activation of caspase activated DNAs. You've learnt about this in the previous lecture. Caspases cleave ICAD, releasing CAD, and then CAD will cleave the DNA between histones. So that will result in DNA being cut up, and that causes the chromatin to collapse and you get nuclear condensation. Now, because the histones have roughly 150 base pairs of DNA wrapped around each histone and then a linker, if you cleave the DNA between each individual histone core, also called nucleosome, you get roughly 180 base pairs of DNA. So if you extract DNA from some apoptotic cells, you see a classic repeating pattern of 180, 360, 540, 720 base pairs of DNA. So in this experiment, cells have been induced to undergo apoptosis in cell culture. So a typical experiment that I will do is put trail on some cells that will induce apoptosis, and then I can follow that every few hours by harvesting the cells and extracting the DNA and then running them all on an agarose gel. And this is just an ethidium bromide stained agarose gel. And what we can see at a certain time point, this is about between 8 and 12 hours, we start to see this high molecular weight DNA here start to be cleaved, and we start to see these repeating units or repeating units of DNA. So this is where we know that caspase activated DNAase has been activated, and we get the classic DNA laddering. So all of this is just a positive control for apoptosis, and it's all based around this observation that caspase activated DNAase cuts the linker DNA here and here, which is why we get the repeating 180 base or 360 or 540 base pair fragments. Now, because we know that DNA has lots of DNA breaks in an apoptotic cell, we can exploit that with tunnel staining. So tunnel staining uses terminal deoxyribonucleotidal transferase, TDT, and this enzyme will add nucleotides onto the end of any DNA breaks. So what we can do is add fluorescent nucleotides onto to the cell and allow this enzyme TDT to add fluorescent nucleotides, and wherever cells have got lots of DNA breaks, they have got lots of well, the cells are fluorescent. This image and this image are the same cells. This is bright field, this is fluorescence microscopy, and about half of the cells here, if you overlaid these two images, are fluorescent. Now you can perform fluorescence microscopy, or you can run them through flow cytometry to check the percentage of apoptosis. You can also do this on tissue sections. So this is immunohistochemistry detecting bromodeoxyuridine labeled deoxy-UTP. Now this is just a modified nucleotide that TDT can add, and you add the enzyme and the nucleotide, you see where it incorporates new nucleotides, and then you detect these nucleotides with an antibody that recognizes the BRDU modified nucleotide, and these cells that are black here are apoptotic, and you can also see they look like they've got condensed nuclei. So that is tunnel staining, can be done by flow cytometry, fluorescence microscopy, or you can do chromogenic immunohistochemistry. One of the simplest ways of detecting apoptosis is by DAPI staining, which I've already mentioned earlier on. But DAPI staining is a DNA binding dye. When it binds to DNA, it fluoresces bright blue. You have to excite it with ultraviolet, and then you get blue light emitted. And here are some normal cells, here are some condensed apoptotic cells, here are some very late apoptotic cells. So this can be done on fixed cells because DAPI cannot cross an intact plasma membrane. So you have to fix your cells with ethanol, or maybe paraformaldehyde. So you have to have dead cells for this, but also it means that you can use tissue sections, and this is a tissue section of a tumour. And zooming into this region here, you can see these very bright spots here. These are condensed nuclei in this hypoxic region of this tumour. So DAPI staining can be used on any fixed cells. It can be fixed cells that have been cytospun, that's centrifuged onto a glass slide, or they can be on tissue sections. For many of my experiments, I like to visualize the difference between apoptosis and necrosis, and I use two different dyes, Hirsch 33342 and Propidium iodide. And Hirsch 33342 is very much like DAPI in terms of its colour. It binds nuclear material blue and fluoresces blue when bound to nuclear material and is excited by UV light. The difference here is this can cross the plasma membrane of a live cell. So I can do live experiments here. I can add Hirsch and that will get into the nucleus of any cell, whether live or dead. And then I can add in Propidium iodide, and that can cross the plasma. That cannot cross the plasma membrane, so will highlight any cells which are necrotic as red. So blue cells are, as we are looking at them, alive. They have intact plasma membranes. The Hirsch has crossed the plasma membrane. The Propidium iodide positive cells are officially dead. And there is a cell that is sort of a pinky colour. It's been stained with Hirsch 33342, and Propidium iodide is just creeping in to the cell. That is a cell that had died within a couple of minutes before I took this photograph. So we can do live cell imaging and watch cells as they undergo apoptosis. And this is just further summarised here. So the apoptotic bodies are just like DAPI staining. You get the condensed apoptotic bodies, either fragmented nuclei or condensed nuclei, or sometimes a crescent nuclei, whereas anything that is large and diffuse is normal, anything that's large and red is necrotic, anything that's fragmented and red is late apoptotic. That's a cell that has undergone programmed cell death, then the membranes have become permeabilized, and then they have properly died. Now in normal tissues, then the late apoptotic cells tend to be engulfed by macrophages rather than leaking out their contents. Now you don't need DAPI and fluorescent microscopy to be able to visualize apoptotic nuclei. You can do it with classic H&E staining or just Haematoxylin staining, and many of you will have done this in the lab where you stain a tissue section with Haematoxylin. Haematoxylin stains the nuclei blue, and you can see condensed nuclei here. So this is apoptosis. It's not quite as good as DAPI staining in that sometimes very small lymphocytes can have nuclei that look like apoptotic nuclei, but it is usable in, especially when you're looking at Haematoxylin and eosin stained sections. If you're looking at those sections just to see the tissue architecture, you can also often pick out areas of apoptosis. Now the other thing you can do is exploit the fact that apoptotic cells express active caspase 3, and we do this by having substrates for caspase 3 which, when they are cleaved, they will release something that is either fluorescent or luminescent. So two assays that I use this short motif DEVD. That's the codes for the amino acids that are the amino acid code DEVD. So obviously V is valine and E is glutamic acid. So that sequence is cleaved by caspase 3. So you can have DEVD tagged luciferin, and luciferin is a substrate for luciferase, and when luciferin and luciferase come together, that gives out a photon of light, so light is related to caspase 3 activity. And these experiments are typically done in a cell lysate. So you get the cells, you lyse them open, you add in your luciferin, your luciferase, and you see if any light is emitted. And this is shown here. This is the caspase 3 vivo glow assay that I use routinely. Here is a DVD peptide on luciferin. When caspase 3 cleaves off the DVD peptide, that releases luciferin. That is a substrate for luciferase, and that releases, when luciferase acts upon this, it chemically changes it to give out light. Now, luciferase-based systems are really good in terms of having a good signal-to-noise ratio. This is something that the biochemist should be particularly interested in. But what we can also do is fluorescent-based assays, which have a much poorer signal-to-noise ratio, but can be visualized by fluorescent microscopy or flow cytometry. So this is one assay that I routinely use. It is a DVD peptide linked to a DNA dye, and whilst ever they are together, they cannot bind to DNA. If caspase 3 can cleave off the DVD peptide, that can release the dye, and the dye can bind to DNA, and the DNA in this case fluoresces bright green. Either later on in the module or possibly in your flow cytometry lectures, you might have come across an Exin 5 staining. I'll not elaborate on it here, but this can also be used to further clarify that these cells are apoptotic. So if you've not covered that yet, you will cover that in a future flow cytometry lecture. Now the other thing that we can do is Western blotting. So again, this will be done using a cell lysate. You take your cells, you induce apoptosis with some drugs, and then you do Western blotting for various different proteins. And this is an image, actually comes out of I think last year's exam paper, and it shows different caspases and in their active form, or the PARP in its cleave form. And it's a sort of experiment that we would routinely do to see whether apoptosis is occurring. So I've got some various questions down here. What would be a good idea to do is have a think about those questions. The answer to them is in this week or last week's lecture. So if you follow those lectures, you should be able to answer all of these questions. Now, all of these are, in the absence of drug is a normal situation. In the presence of drug, you're getting a pro-apoptotic response. So we're detecting proteins that are associated with apoptosis, but hopefully you will be able to figure this out, because it's exactly the sort of problem that you're likely to get faced with on your exam. And here is another example for you to have a look at. So from this, you should be able to figure out what is happening in terms of these drugs. So this is trail, and I'll just tell you that VAD-FMK, that is a chemical that we use, which is known to be a caspase inhibitor. Not telling you which one, you should be able to figure it out. So in the live session tutorial, I will go through both of these Western blots and make sure that everyone's happy with the reasons why we are seeing those particular effects. Now this is a summary table of all of the apoptotic assays that I've used as part of my research. Some of them I've talked about today, some of them you'll learn about in your flow cytometry lecture. So I've mentioned H&E. It can't be used by flow cytometry, obviously it's light microscopy. Very simple, compatible with tissue. You can also do that on cells from cell culture. DAPI is on fixed cells or tissue because it's permeabilized cells, whereas Hirsch and PI is limited to live cells, where PI, as in red cells, are dead. You can do DNA laddering. This tends to be require a high proportion of typically cultured cells. This is the best situation to do this experiment. Get some cultured cells, extract the DNA, run it on the gel. Sub-G naught analysis is directly related to your flow cytometry lecture. You should have learnt about how to differentiate between G1S, G2. Well, sub-G naught are smaller than G naught or G1 cells. So you should have learnt about that in flow cytometry or in a future session. Again, same with the Nexin 5. If you've not heard about it yet, you'll hear about it shortly. And then we've got our caspase activation assays. I used one called NucView 488. That's the fluorescent green substrate that caspase 8 cleaves, releases the green substrate, and that goes and binds the nucleus, and we can do that by flow cytometry or fluorescence microscopy, or we can use the luminescent version using a plate reader that detects luminescence. Okay, so that summarises all of the assays, all of the likely assays, and you will cover some more as we go through the module, and especially when you do specialist flow cytometry lectures maybe in semester 2. So the sort of question that I could ask you on the exam is, here is an experimental situation, propose the most appropriate apoptotic assays that would distinguish between apoptosis, normal and necrotic cells. And that's a sort of question that I may ask you to answer. Okay, so that's the end of this session.